more of it dissolved in plasma, right? Because it's more soluble. There's less of it in red blood cells, so it's not hidden. Okay. Oxygen's hidden, right? It's in red blood cells. It's hidden. So for that reason, it's easier to monitor CO2. And the other is, what do you think it's easier to fine-tune and to really regulate? Something that varies under rest between 40 and 100, or something that varies under rest between 40 and 46? This is much easier to regulate on a finer level, right? There's less to take care of here. It's only going to vary between 40 and 46, so I can really fine-tune that. This one varies over a much broader level, so it's harder for me to regulate at a finer level. So what we regulate, or what we really monitor, is carbon dioxide. Now, you can get away with that because if CO2 levels are low, what does that mean O2 levels are? High. So I should slow my rate of breathing down. If CO2 levels are high, that means O2 levels are low, and I should do what? Increase my rate of ventilation. Does that make sense? So we can monitor one or the other. It doesn't matter which one, because they're correlated with each other. If O2 levels are high, CO2 levels are low. If CO2 levels are high, O2 levels are low. So we can monitor CO2. So these are chemoreceptors. So we have chemoreceptors that monitor carbon dioxide. And they monitor carbon dioxide in two places. They monitor it in cerebrospinal fluid, and they monitor it in blood plasma. Okay. Cerebrospinal fluid, these are called central chemoreceptors because they're monitoring it within the central nervous system. These are called peripheral chemoreceptors because they're monitoring it in the peripheral nervous system. Does that make sense why we just call the two things? They're kind of the same, right? But they're just called two different things. Now, this is actually going to be probably monitoring cerebrospinal fluid around the brainstem. So we have